Okay. Hi, good morning, everyone. Uh, Sorry, I don't have visibility, so is everyone in yet? Maybe we'll give it around 1 or 2 minutes for everyone to filter in, and then we'll start the webinar soon. Okay. So I see quite a number of people have joined already. So let's begin. So good morning, everyone. I'd like to thank you all for joining our webinar for today. So this will be in collaboration with PacBio. And the topic for the day will be to unlock the full potential of genomics with PacBio HiFi long read sequencing. Now, before we begin, I'd like to go through a few webinar guidelines. So all attendees are automatically muted upon joining and should remain so throughout the entirety of the webinar. But no worries, because there will be a live Q&A session conducted after the presentations. So in case you have any questions during the speakers' presentations, feel free to type your questions in the chat and we'd be happy to answer them towards the end. Furthermore, this webinar will be recorded, so you can ask for the recording after the webinar as well. So you can just have a look and watch it back. And before we go to the speaker presentations, I'd like to just share with you guys a bit about Novogene and who we are for those of you who don't already know us. So Novogene is a global leader in multi-omic solutions. We empower discovery and innovation across genomics, transcriptomics, single cell and spatial transcriptomics, metagenomics, and even proteomics. We have one of the world's largest sequencing capabilities with a strong network of scientific expertise and a commitment to our client success. We have 10 laboratories globally with over 60 high-throughput sequencers and over 2,000 employees. We process over 3 million samples a year for over 7,000 customers. So I hope that gives you a sense of our scale. And speaking of scale, here are our 10 laboratories worldwide. We are located in the UK, Europe, America, Japan, and China, where our HQ is. And where this webinar is being held is the Novogene Singapore facility. Now, we serve over 10,000 projects annually from 15 countries across the Asia Pacific, Middle East, and Africa. So, the logos you see below are just a little look into the partners that we have and the clients that we serve. So Novogene will have the full range of multi-omic services. If you can see from left to right, genomics and transcriptomics are our bread and butter. We have microbiome analysis, epigenomics, and more recently, metabolomics and proteomics services as well. So if you'd like, feel free to take a screenshot, have a look at the services we have. And in the case that you're thinking of planning a project, do contact us and let us help you along the way. Now, I'd like to introduce our first presenter for the day, Mr. Wilson Cheng. So Wilson is a staff bioinformatics scientist on the PacBio Asia Pacific team and has been working with PacBio long-read sequencing data since 2012. He specializes in human whole genome analysis and clinical applications of HiFi sequencing. And before joining PacBio, he's contributed to the development of some WGS workflows as well. So Wilson earned his degree in bioinformatics from the Institute of Biomedical Informatics at National Yangming University, and later joined the drug design project team at UCSF in California. So without further ado, I'd like to bring Wilson on to the talk, and I hope you find his presentation informative.
Right. Yeah, thank you, Kim. And let me share my screen. Can you see my screen now?
Yeah.
All right, cool. Yeah, let me turn on my laser pointer. Okay. Hold on. Okay, I'll just start. Yeah. So screen check and mic check again. All good?
Yeah, all good, Wilson. Oh, Wilson, you froze for me. Are you still there? Oh, hold on a second. I think Wilson needs to rejoin. Give us a moment, and I think he might be having some technical difficulties.
Sorry, I got some internet issue. Can you see my screen now?
Oh yeah, you might want to start from the beginning, Wilson, because I think we lost you quite early on.
Oh, sorry. I don't know what's going on. I
Yeah.
guess something wrong with my Wi-Fi in the hotel. Sorry about
Yeah,
that.
no worries, no worries.
Yeah, so sorry. Yeah, I'll get back. So I just again, I'm Wilson, so I pretty much covered the latest progress that in genomic research using paper HiFi sequencing. So as a biometrician, I will cover in some of detail of the data and let you know why HiFi is so special. So I will start with the recent review paper published on February this year. So that summarized the impact of HiFi sequencing across genomics. So the paper title actually you can see here, Long and Accurate, How HiFi Sequencing in Transforming Genomics. And the highlights in here that HiFi can cover different kind of variation, TDC assembly, and even metagenome assembly, and some complex region like rDNA and the centromere annotation. So pretty much summarize everything that the power of HiFi. I encourage you guys, if you have time, you can take a look on this paper. And then I will let you know the background of the HiFi sequencing, what it is, and how we generate it. So we start with a high molecule weight double-strand DNA, and next we will ligate the adapter to both ends of the insert DNA. And this will form a circular form of the double-strand DNA. And once this adapter anchored to the polymerase in the small cell, it starts generating the sequencing signal. And it's a real time, so we'll collect the signal directly and do the base calling and instrument. Yeah. And then because it's circular form, so pretty much we'll sequence the DNA molecule several times. And as you can see here, there's some red dot, and the sequence. So they're pretty much the sequencing arrow. And because the sequencing arrow are random, so we can pretty much give you the consensus of this sequence and will wash away of the arrows. So thus we can give you the high quality consensus data called HiFi reads. And most of our rate pretty much will give you over Q30 accuracy and can be up to 25 kb. And if you use a different site selection platform, maybe you can up to 50 kb sometimes. So now you know HiFi sequencing provide long and accurate reads of native DNA. So it delivers a real length from up to 25 kb and extremely high accuracy. And we don't have system bias, so we can give you the complete even coverage. And it's a native molecule, so we can detect the epigenetic signal at the same time. And average quality of our data pretty much over Q30. So here's a landmark example of what HiFi sequencing has made possible. So we launched HiFi reads in the late 2019. So the telomere to telomere consortia quickly adopt to the technology. And shortly after, they published the first ever complete human genome assembly, the CHM13 reference, including the T2T telomeres and centromeres, which had never been fully resolved before. And this quote by NIH Adam Phillips even said that the all-star of this assembly has been PayPal HiFi. And just pretty much this shift paradigm of the genome assembly. So pretty much you can see a lot of TC genome by HiFi right now. So we are not just doing the typical TUT right now. We are moving to a pan-genome level. So this is just, I won't go detail. I will just highlight that, let you know that we are doing some pan-genome project right now. And not just human, but also the plant genome. So the resolution HiFi is powerful enough to handle even the most complex plant genome. So this is an example from hexaploid genome. So you can see this IGV view of the assembled context. The resolution of so high is like we can assemble 6 haplotypes directly with HiFi. So with this powerful power of the HiFi ability, you can see pretty much we involve a lot of big, large project like Earth's Biogenome projects, Darwin Tree of Life, Beta 1K, BGP. So I won't go detail here. Our next speaker, Liu Rui, will go deeper into some of the PS case studies. So back to the human reference genome. Here we have direct comparison between the short-read NGS and HiFi-based TUT data. So on the left, you can see here, there are a lot of gap right here. So they're located some of the hard to map regions like homologous regions or repeat regions. And the right-hand side is a HiFi sequence over. We give you the full resolution of the chromosome information. And not just the DNA itself, we will also give you the epigenetic signal like 5MC methylation and also the 6MA methylation. So what's going to happen, like what's the missing part in the NGS? So here is the NGS. When you align your data to the typical human reference genome, there's a missing part. For example, this is the STRC gene located in chromosome 15. And it's well known that it's a clinical relevant gene and have a nearby pseudogene. So this homologous region makes it hard to be mapped precisely. Like you can see the bottom panel here shows the short-read Nova-seq data. So you can see there's some mismapping region and non-mapping region. And with a low accuracy and unmapped gaps here because the pseudogene is on the other side. Whereas people HiFi on the top, you can see here, using HiFi reads from Revio system, you can see not just fully covered gene. The solution of HiFi is so good, so we can actually phase the reads into the different types and different haplotypes and confidently detect the variants. And remember, we are sequencing the native DNA and we can get you the methylation signal at the same time. So you don't need to perform the bisulfite treatment or bisulfite sequencing, another round of analysis. All you need to do is just click on a button in the IGV view, and you can see the 5MC signal with the phasing result directly. And here's another powerful example of what HiFi can do. So phasing made all difference in this case. So it helps us resolve what traditional sequencing couldn't. So standard short-read sequencing couldn't explain the disease. It missed the full picture in this case. So you can see about the HiFi, people HiFi sequencing data, we identify full pathogenic variants in the same gene about 10 kilobase apart. And most important, we were able to phase them. So pretty much 2 variants are from 2 different alleles. So which means that both allele of the genes are mutated. so cause of disease. And as you know, this we call it a heterocompound heterozygous mutations. So with this, HiFi can give you the ability to resolve this kind of case easily. And there are simply too many impactful cases to list them all. So I won't go detail. I just let you know we have involved a lot of large national biobank project or disease project from Children's Mercy, over 1,000 genomes, SOF-RD, Reborn, and Huntington's Alpha. And with this, we have collected a lot of information. We found that actually many of these unresolved cases, a lot of answers lies in the structure variants. And we know that we are good at structure variation, but not just typical large SV. Also, we try to interpret some of the complicated cases, like inversion of something else. So we try to develop a new type of the variants. So not just typical NCP, SMV indel, and structure variation. We also try to find some hard to resolve, difficult to resolve, and the clinical relevant regions, such as some repeat expansion region or some segment duplication region. So the first category I want to highlight is the tandem repeats. So we have found that a significant portion of the pathogenic structure variants are falling within tandem repeat regions. So these regions are hard to resolve with short reads, but HiFi sequencing provides both the relance and accuracy to resolve them easily. So I want to highlight this is paper. So this paper published early last year. So just give you some information that it can be not resolved until we provide the solution of this data. So you can see here, actually you can find the patient have the two allele information right here. So one allele have multiple GTC expansion and cause of disease. And with this, as to let you know, we have power to do this because we have this tool we call the TRGT. So the tool is a tandem repeat genome typing tool that we published, I think, early last year as well. So the tool is easy to use. Pretty much just give you just input the alignment band and some repeat definition. And here's the FMR1 expression definition file in here. So the output will give you not just the motif number, but also two-phase methylation information. The reason that we put the methylation signal right here is because we found a lot of hypermethylation region related to the pathogenic region. So this will give you a full solution of the tandem repeat expansion information. So next, let's move to another types of repeats, second multiplication, or you can say the homologous regions. So there are around like 7% of this kind of segmented duplication regions. So some of them are pretty large, pretty long, and they're highly homologous regions. So it's hard for the short read to map to this kind of region and give you the resolved information. So I will use SM1 as an example. So what makes this region especially difficult is that because SM1 and SM2 are over 99.9% identical. And they're pretty much 3 kb in length and differ by just a few bases. So the most well-known difference between S1 and M2 as located in the exon 7, there's only one base difference on the first base of exon 7. So here you can see the mapping results across a different platform. So here's the SM1 gene, and in our panel, you can see the show read data. So we're over 84% of reads mapped with a MAQ0, meaning the aligner could then confidently assign them a clear sign of the mapping ambiguity in S1 and S2 region. So in contrast, HiFi WGS show in the middle reduced this dramatically. Only 2% of reads have MAQ0, and the misaligned read dropped to 90%. So we developed a tool called So this will give you the tailored mapping results. So you can see here, this mapping band gives you a four-phased category of the reads. So you can see here, the first one's SM1 haplotype 1, and the second one's SM1 haplotype 2. And we also give you the information, the SMN2 information region as well. So this is a highlight of the paraphase that have been used in we so-called HiFi Cell Consortia. So we have HiFi cells in APAC right now, and also we have it in the AMEA. So what we do is pretty much we sequence a bunch of the patient samples, and in the first tier of the report, they found that a lot of this kind of variants are located in the so-called segmental duplication region. So Prevace really helped them to resolve this kind of the variation in these genes. So with this project, we are now not just detecting just the typical homologous genes, but we also can increase up to the 160 gene families. And we have published paper earlier this year. And it's free to download, and you can use it from our GitHub. So overall, you will want to know that what's the sequence depth you need, right? So this is a performance of the PacBio and other platform, and we're using different software to give you the information of Recode, SME, and Intel. So out of three platforms, PacBio give you the most accurate variant cost. And then we want to know the sequencing depths, right? So I have shown you guys this slide before. So we'll give you the additional explanation with HiFi. And the good thing is that you don't need really high coverage. Some of them only use 10x coverage, some of them use a 25, some of them use 30. So we do a typical titration way to detect, to see what's the performance of a HiFi. So with a 20x of HiFi, and we use a different platform to compare. You can see here, HiFi gives you the fewer errors compared to the benchmarking data. And then you can see titration right here. So they are the nanopore, 60x nanopore, and 20x HiFi here. So we think just around 20x HiFi will give you the most accurate SMV and Intel call compared to other platform. And SV as well will give you a more accurate SV call compared to other platform. And we try to do the same with the methylation detection as well. We also do titration from 25x to 5x. So around 10x give you the pretty much quite reliable data compared to the short read data. So all of them, we combine them all together. We have different types of the tools, not start from the Snip-Indel certification, and like I said, the tandem repeat and segment duplication. And we support the phasing, and we support combinatorial variation. And the WGS data, we can call the PGX and HA typing directly. And you know that we have methylation information at the same time, and we can call the mitochondrial variants as well. So all of them combined together, we come up with a pipeline we call the WGS-WDO pipeline. So when we say pipeline, means that it's a command line tool that you just type one command, it will give you all the information, all the output from all data that list in previous slide. And it support the cloud, so it gives you the second analysis result, and we combine our long reads colors DB control database, help to figure out a part of the non-variants. And we cover with the Gennomics and Genix. Say if you want to have a tertiary analysis report, I think the typical NGS or Gennomics Genix tool can help you to give you some ACMG recommendations. So this pipeline has been used in the worldwide human genomes. What I highlight here is that they are all happens in APAC, including the Japan Genome Project, Korea Performance Genome Project, PRECISE, Asia Pangenome, Chinese Pangenome Consortia. And next, I will cover the RNA part, isoseq. Okay. So the slide highlights the limitation of short reads in resolving isoform compared to a long reads. So in RNA sequencing, short reads often struggle to provide a complete picture, whereas long reads capture the entire transcript in one piece. So typically, the read length, the gene is around only 2 to 4 kb in human genome. So for HiFi, it's easy to cover the full gene of the mRNA, right? Whereas short read, you need to assemble first, and sometimes they give you the round assemble or different round facing result with the short read data. So it's pretty straightforward. We can give you the full picture of the isoform. So it's a pretty much that straightforward that use this isoform to annotate the genome. So we have help update a lot of different kind of the mRNA database along the world and involve a lot of plant genome with HiFi assembly results. And not just a typical gene annotation, we can also help to resolve in a lot of disease, especially cancer. So I will give you some idea why. So to sequence the cancer genome, we do isoform. What we do actually want to increase the sequencing throughput first. So you can know that the RNA-seq, the DNA gene length is around only 2 kb, whereas HiFi can give you around 20 kb length, right? So what we want to do is that we want to concatenate this gene into one HiFi read to increase the throughput. So this will give you the kinetics solution. So we have published this solution, I think, a few years ago. And then we have give you the kit. So pretty much you can get a kit and you can get a kinetics solution for the bulk iso-seq and also the single cell iso-seq. So beside the sequencing kit, we also provide another solution. The same as a pipeline, it's like one command line, or we have a UI, you can click a button, then you can give you the result. And some third-party tool can support the character analysis tools. For example, like Pigeon, Squanti give you the annotation of the gene and give you the fusion gene information as well. And this Kana and Scampi give you the single cell information. So here are some examples of the output, direct output from the pipeline. For example, this is a way that you can find the novel splicing form with oncogenes. So in this paper, pretty much they found a lot of oncogenes. They have a lot of a novel isoform. The novel isoform means that you can see in the middle of the picture. So there are different colors of the aligned isoform here. So all the reads are from HiFi, and when they align to the reference, you can see part of them are full splice match, means that they're 100% the same as the non-transcripts. And there are some of novel in catalog, means that this splice form, these exons are known, but this kind of splicing forms are new. And novel non-clinical means that we can find some novel exon in the novel splicing forms. And this is not just a typical structure variation, it has a real functional consequences. About 30% of novel isoform affect the conserved domains, and the 35% are predict to alter protein localization, which can impact oncogene signaling, therapy resistance, and the immune escape. So this kind of isoform level resolution has real implication for drug design and immunotherapy. And another way around is that we can give you the isoform level expression information. So for example, this is a tool called Talon. So it's a third-party tool, but you can just combine the different transcriptome output from PacBio HiFi. For example, I have a different type of the cancer or different part of the cancer tissues. And then you can see that in the different types of the data, the types of sample, you can see that different cDNA can be detected, and we have the isoform expression value in here. So this can give you find out some like tissue specific or cancer specific isoform can be found in your data. And the good thing is that it can be the single cell level, like I said. So the output of the our pipeline can directly import into the 10x analysis pipeline, So you can still give you the typical single cell analysis results. And another one is the fusion gene. Like you can see here, there's a mapping result of the reads, and pretty much you can see there's a combine and a different two transcript. And again, this kind of fusion gene can be the single cell level. So overall, that HiFi can give you the diverse type of applications from targeted panel, whole genome, or variant detection, DML assembly, or RNA level. And we have standard sample preparation kit, lab preparation kit, and sequencing platform. And we also give you the standard analysis solution. So pretty much everybody are using the same analysis pipeline. And with this, I just want to let you know that Pearl HiFi, like mentioned previously, multi-omics. So pretty much the throughput of HiFi is getting higher. So one cell can not just give you the genome information, but also give you the epigenome information, even a chromatin methylation signal. So you can find all the information in one time, including gene expression. And with that, thank you for your attention.
All right. Thanks, Wilson. Thanks for the informative talk. I'm sure there'll be questions for you towards the end of the presentation. Now if Laura can start sharing her screen.
Can you see my screen?
Not yet, Laura. Okay, yeah. Your screen's available now. Okay, so just to introduce Laura a little bit, she is our senior product manager at Novogene AMEA. So Leroy attained her master's degree in biotechnology from the Hong Kong University of Science and Technology. So she joined Novogene in 2020 as a technical specialist, where she mainly provided services for researchers across various fields, including microbiology, disease development, and breeding programs. So her responsibilities were mainly to assist researchers in finding NGS solutions for their research projects, and solutions for bioinformatics analysis. Leroy has since transitioned into the role of product manager in 2023, where she primarily focuses on studying market trends and applications of long-read sequencing technologies, PacBio included. So with that, yeah, Leroy, you can continue on to your talk.
Okay, thanks for the introduction. So I will start today's introduction on the Novogene sequencing service using the PacBio HiFi library system. So here you can see is an overview of the Novogene sequencing service using the PacBio Revio systems. We could offer the service from the genome sequencing, transcriptome sequencing, microbiome-related sequencing, as well as the epigenetics and primary libraries. And all these sequencing were performed on the latest Revio platform. And I think most of you might know one concern about the PacBio long-read sequencing is the cost. Well, with the several continuous updates from the SQL2E platform to Revio platform, and now the latest bar chemistry, the data output per cell is increased significantly. And this increase has also lowered down the sequencing cost per gigabase, and it making the PacBio sequencing more accessible than ever before. So here are some of the testing data from the Novogene. So here you can see one of our mammal samples can reach to 144 gigabase using the latest Revio platform and the Spark chemistry. There is about like around 37% increase compared with the previous Revio and previous chemistry for about 90 gigabase. Also, in comparison, the previous SQL2E system can only generate for around 15 to 20 gigabase HiFi data. So there will be a very huge increase on the data output per cell. So here are some of the detailed information of these 3 testing data. You can see all this data of very high quality. The read longs is above 15 KB, and all this HiFi read is over Q30 with the quality. And here is also the HiFi data read longs distribution and the data quality distribution. Also, I want to mention the on-instrument autograin has been updated as well for the improved 5MC detection accuracy. So these are also the testing data from the Novogene. Here we show the predict possibility of the 5MC of different samples. You can see compared with the previous version, there is a huge decrease of the low quality data, which means the accuracy of 5MC has been increased. And thanks for the launch of Spark Chemistry, the Novogene can offer a promised data output per cell as well as a promised HiFi data accuracy. So here we could offer human samples a minimum of 130 gigabytes HiFi data per cell, while for plant and animal samples, you could get around 110 gigabytes per cell. However, for different species, the actual data output may vary. So if you are interested in the sequencing service, you can contact us for more details. Also, I think it's worth to noting that the increased data output is actually attributed to the optimized enzyme activities of the Spark chemistry. And this actually can address the problems or issues from the many tricky species, such as the aquatic species. This kind of species might contain like impurities or contaminations and will cause the early termination of the enzyme. While with the latest Spark chemistry, the data performance of this kind of species will be better than before. And here is the analysis that we could offer containing the variant detection, the de novo assembly and annotation, the methylation information analysis. So here let's move on to today's main topics on how to get a high quality de novo assembly from the sample preparation to the insights. So I think most of you might be interested in the de novo assembly and annotation. So I will start from this section first. So what is the de novo seq and why it is important in many research areas? So de novo seq means to sequence a novel organism like in a reference genome and to construct its genome map. So usually we can think of the de novo seq as to putting together a puzzle. So when you putting all these reads together, you will get a genome map finally. And this final genome map can further be used for the analysis. So I think most of you might be familiar with the resequencing. Through the resequencing, we map the reads to the reference genome, and we can perform the variant calling. And these results can further be used like GWAS analysis for the association between phenotype and genotype, the BS analysis to identify the genetic markers associated with a specific phenotype, the population genetics, such as the allele frequency dynamics and the evolutionary forces. So how do we start the analysis for a species without a reference genome? So in this case, we need to start the de novo seq first and to construct the reference genome, and the following analysis can be applied later on. Besides, the individual genome construction is also important because each individual genome is different, and it will help us to understanding the genetic variations. So we can use like the comparative genetics analysis or the pangenome analysis for further analysis. So usually the comparative genetics focus on the comparison of genomes across different species. You can contain the information such as the species divergence, while the pangenome is mainly focused on the studies within the species. So through the pangenome analysis, you might get information like the core genes, the genes present in all individuals, and you can also get the dispensable genes, the genes only present in several individuals within the species. So in summary, the genomics is quite important because it can be applied in several different research areas such as species evolution, genomic variations, and genetic diversity. So here is one of the examples from our customer. The Novogene offer the library preparation and sequencing service and part of the analysis service. So this study mainly focus on the reference genome construction and the population genetic analysis of the piece. So we know that consumpable peas is one of the most important crops globally, and the peas can be classified into several different types, the green pea, vegetable pea, and the vegetable pea can further classify into the vegetable seed pea, the pod pea. So, however, this study mainly focus on the analysis on the genetic differentiation between the green peas and the vegetable peas. They also developed the candidate genes that is associated with the crucial agronomic traits. So here is the research pipeline of this study. You can see that the study starts from the assembly of the J1 number 1 to construct the reference genome. They get a chromosome level assembly using the PacBio, HiFi, and HiFi technology. So after get the reference genome, they further perform the comparative analysis between the J1 number 1 and J1 number 6. Later on, the population genetics and the GWAS analysis was performed against the reference genome we get at the beginning. So through these two analyses, they further identify around 200 candidate loci associated with 57 important trees. So this is the general analysis pipeline for the genomic agriculture genomics. So here are some of the analysis results in this paper. So firstly is the comparative analysis. So here it shows the syntactic and non-syntactic regions between the two different assemblies. You can see the green part refers to the syntactic regions, while the other regions labeled in other colors refers to the non-syntactic regions. For instance, the yellow one refers to the inversion, while the blue one refers to the duplication. So on the other side, they also perform the population structure analysis. So this analysis involves in total 340 samples, and 237 of them are the newly resequenced, and 77 of them are from the published database. So they first performed the phylogenetic trees. So you can see the result here. And all these samples are divided into 3 different groups. So the group 1 is regarding the wild progenitors, and the group 2 is containing some of the green pea and some of the vegetable pod peas, and the group 3 is regarding the vegetable pod peas, vegetable seed, and some of the Aphylla type pea. And the PCA analysis also shows the similar patterns. And through this population analysis, they finally get the conclusions that some of the genomic regions with the largest difference between group 3 and group 2 are located on the chromosome 1, chromosome 2, and 7. And these genes are mostly associated with the symbiotic fungi and the lipid transport. So lastly, they performed the GWAS analysis. So the 237 samples are further phenotyped, and they select for around 50 important agronomic trees for the further GWAS analysis. And here it shows some of the analysis regarding the phenotypes and the candidate genes. So, for example, this regarding the stem length and the related candidate genes. So there is a distribution of the lossy associated with the agronomic trees across the genome. So here is the chromosome from 1 to 7, and here is regarding the agronomic trees information such as the seed weight, the seed color, and some other trees. So when you decide to start using the de novo seq in your research, so you might wondering what is the suitable platform for assembly, how many reads is enough, and what pipeline can be used to get a high quality genome. So you will find this information in the following slides. So firstly, there are actually several different level of context assemblies. So firstly, the reads can be aligned together to form the contigs, while these contigs do not have any sequence or the directions. So these contigs can be further joined and oriented together to form the scaffolds, and this scaffold will be further assigned to each chromosome. However, in this stage, this chromosome still contains many gaps, so we need to use much longer reads to fill these gaps and to get the complete gapless chromosome, which we also call the telomere to telomere assembly, the T to T level assembly. Here we also show some of the recommended sequencing strategy for a common diploid species. For the context level, we recommend 30 to 16S HiFi reads, while combining with the 100X HiC data, you can orient the context to the scaffold level and further assign to the chromosome level. So with the combining of the ultra long data, especially the reads over 100K, so we can fill the gap to get the T to T level assembly. So you can see for each of the level of the assembly, the HiFi data is required. So why HiFi long read sequencing is quite important in the de novo assembly. So here are the reasons. So firstly, you can imagine when we're putting together a puzzle, the larger the pieces, it might be much easier to putting the puzzle together. So it is actually same with the de novo assembly because like the GC-rich region, the repetitive regions are quite similar with each other. So for the short read sequencing, the reads are actually similar with each other. You are unable to locate the right positions in the genome, while for the long read sequencing, over than 50K, the reads contain more information except the repetitive or the GC-rich regions. So it will help you to locate the positions in the genome. And the long read can also help to bridge the gaps left by the short read assemblies. Also, compared with like the nanopore platform, the HiFi long read sequencing can offer a very high quality assembly. The base accuracy is usually above the Q30, while in general, a nanopore platform can generate reads around like Q20. Yeah, so it's quite important to use HiFi data to construct the backbone structure of the genome. Also, before the genome assembly, we also recommend 15x short-read data for the genome survey. It will help you to estimate the genome size, the heterozygity, and some other useful information of the genome. So here is our general analysis pipeline. The analysis will start from the genome assembly using the HiFi exam. And after you get the sequence, the assembly sequence, we will perform the annotation. Through the annotation, you will know which region is the repeat sequence, the gene structures, the gene functions, and the non-coding RNA information. So on the left is the analysis pipeline of the de novo assembly. So at the beginning, all these reads will be clustered together. So for bases with very quite low frequency will be identified as the sequencing arrows, while the other bases will be identified as heterozygous allele. And after the correction, error correction of the reads, all these reads will be aligned together using the overlaps. And due to the presence of the heterozygous allele, there are commonly two different pathways. If you have like complementary data from the HiFi technology or the parents' data, we can generate the haploid type assemblies. So if you do not have these supplementary data, we will only deliver the primary assemblies. Regarding the annotation for each of the annotation, the repeat, gene, and non-coding RNA, we will combine different prediction methods such as the de novo prediction, homolog prediction, and all these results will be combined together and delivered to the customers. Well, regarding the gene function annotation, we will mainly use the database function annotation method. So regarding the T2T assembly, the pipeline is slightly different. We will merge the data from the different platforms since different platforms always have different features, and these features might have a preference, a sequencing preference of the genome. So it will be more likely to get a complete genome by combining these different sequencing techniques and different data. And that's the reason why we do not use the single platform. So with the single platform, it commonly can be used for small genomes instead of the large genomes. So here are some of the detailed analysis pipeline. So the HiFi reads and the ultra-long reads will be assembled separately, and we will merge the data together. After get the draft genomes, the ultra-long data, especially those over than 100K, will be used for the gap filling, and then you will get a finally T2T genome. Here are some of the project information, project experience of us. So here is the genome size, the chromosome number, the gaps number, and like the telomere number, centromere number. So this also an example from the paper we just mentioned. So they use the Illumina data for the genome survey, the PacBio and HiFi technology to get the chromosome level assembly. The ExoSeq and RNA-seq data here is useful to help with the annotation. And here is the genome assessment result. Through these parameters, you can see that the genomes is of a very good quality. And on the right here is the HiFi heat map and the circles plots of the genomics. So from the outside to inside is the genome locations, the gene densities, T densities, NEAT densities, etc. So next we will move to our last session. So we just mentioned the analysis pipeline from the assembly to the annotation, and we also share some of the examples on the application. So when you start the experiments, you might wonder what is the qualified DNA sample that can be used for the PacBio HiFi long-read sequencing. So here we will show you some of the our experience. So at the moment, the main challenges is regarding the DNA input and the optimal high molecular weight DNA quality. So usually for a Illumina sequencing, we usually use like around 200 nanogram, while for the HiFi long-read sequencing and other long-read platforms such as the nanopore platform, we require from a range from the 3 microgram to 10 microgram to construct the library. So the input is much higher than the Illumina platform. The another challenge is the optimal data quality. So to get the HiFi long read, firstly, you need to ensure the fragments of your samples is long enough to do the HiFi sequencing. So here we recommend some keys we have tested internally that can offer a very high quality, high molecular weight DNA extraction, as well as the PacBio extraction kit. And here are some of the important parameters regarding the amount, volume, concentration, and purity fragment size that we recommend for the PacBio HiFi long-read sequencing. So there are several important parameters. The first one is the fragment size. For qualified fragments, we recommend it to be above 30K because this is the most important quality to ensure you can get the long reads first. And when you do the internal testing, we recommend you use the phosphoryl gel electrophoresis instead of the traditional one, because you can see the comparison here. When using the traditional one, it quite not suitable to separating the high molecular weight DNA. Well, when you're using the phosphoryl gel electrophoresis, you can see the real fragment size is around 40 to 50K. So the second parameter is the sample amount, because this will actually directly affect the library quantity and the subsequent data output. The purity is quite important as well, as we just mentioned. The impurities might cause the enzyme early termination, and this will cause the reduce the data output. So a very pure samples is quite important to ensure the data quality. So instead of the standard way, using the 3 microgram library preparation, the Novogene also offers a low input method for human samples, starting from the 1 microgram samples. Here are some of the testing data with from a range from the 500 nanogram to 1 micrograms. And here are the data output and the read longs. You can see all of these samples are of very high quality. All these reads are above 50K, and the data quality is above 30Q38. And I also want to mention that the PacBio offers ultra low library preparation kit. However, if you want to generate a very good quality data, we actually do not recommend to using that kit because it relies on the amplification of the DNA. So during the amplification, the fragment size might be lower than expected. So in this case, the low input method from 500 to 1 micrograms is using the optimized workflows without any amplifications can generate a very qualified data. And here we also compare the low input samples with the standard sample. The H7, H8 refers to the low input one. The S1, S2 refers to the standard one. You can see the statistic results of the SV detection. So the number of SV detected by the low input sample is slightly fewer than the standard one, but they are actually still within the similar level. So so it means that our low input methods is qualified and can be used for the standard analysis. So this is all about today's introduction on the Novogene analysis pipeline on the de novo seq and sample preparation. Thank you.
Okay. Thanks for the talk, Liu Rui. And yeah. So I'm just looking at questions in the webinar chat. We don't have too much time, but for now, I think we have a questionnaire that's on your screen, and we'd be really grateful if you could just take 1-2 minutes of your time to answer the questionnaire, and we'll move on to the Q&A. So we'll give you some time to type your questions in as well. Okay. Right. So while everyone's answering the questionnaire, Liu Rui, I think this question goes to you. So one of our attendees asked, does de novo sequencing have any criteria for selecting individuals? Since the samples are quite small, we need to pool quite many to reach sufficient DNA quantity. And would this impact assembly?
Okay, so actually, in our general analysis, we actually recommend to use the single individual to construct the genome because, as we just mentioned, each individual genome is quite different. So if you pull different genomes together, it might be increase the difficulties to get the assembly. However, I understand for some of the small species, with very small genomes, it's quite difficult to get enough input samples for the PacBio HiFi library sequencing. While in this case, we could pull all these individual together to first ensure we can get enough data from this sequencing. However, considering in this pool, there are several different individuals and the genome might be slightly different with each other, so the final genome size might be slightly higher than the estimation. So this is one of the issues. While the assembly can be performed yearly, just there might be several issues, as I just mentioned, regarding the analysis results. Yeah.
Okay. So I hope that answers your question. Thank you, Liu Rui. And I think we have time for maybe only one more question. So I'm going to pass this on to Wilson. Someone asked, saw Wilson's slide mentioning that WGS can be done using saliva. If the customer wants to get 30 times coverage, do you have any recommendation on how much raw data is needed, since saliva might contain bacterial genome contamination? Or do you have any suggestions?
Yeah, as a bioinformatician, I have some. I know that we have a specific protocol that can treat for the saliva samples. So it's in our homepage. I can send it out to you, And we have a demo data set as well. So pretty much the data is sufficient enough. 30x is sufficient enough for the typical WGS code. And yeah.
Okay.
So 30x should be enough. And the combination rate is pretty low. Yeah.
I see. Okay. Thank you, Wilson. I hope that answers your question as well. So I think we have to bring this webinar to a close since we're running a bit late on time. So you can contact us anytime using the QR codes above. If you have any further questions at all, feel free to write in an email to us, and we will try to answer your queries as best we can, and hopefully we can help you with your PacBio project. So with that, I want to say thank you everyone for joining, and we'll see you all next time. Thank you.
Thank you.
Thank you. Bye-bye.